Great. Thanks so much for inviting me to give a talk. I'm gonna highlight work from Ching, Lin, and Adika in the lab. And I run a joint lab at Janelia with Marius Pakisari. Sorry, I have a bit of a cold, so hopefully you can still understand me. And let me just, can you see my pointer? Can you see my mouse?
Your mouse, yeah, it's not a pointer right now, but the mouse I think is good.
Okay. Yeah, okay, I'll leave it this way. All right, cool. So in our lab, we actually don't do EFIS. We do calcium imaging recording. So this is an example recording of dual plane imaging from primary visual cortex and higher order visual areas. And this is just the raw data. All I've done here is motion register it. And so we can get a lot of neurons simultaneously with calcium imaging. Uh, and at the same time, we can also do visual tasks with the mice. So yeah, calcium imaging is amazing in terms of yield and stuff. And it's a bit easier than doing EFIS. So I think all of you here in the course are very brave. But we have this trade-off like I'm showing here. If we're recording more neurons, we end up going slower. So it changes the sorts of questions you might want to ask. Like you don't want to ask specific spike timing questions with these kinds of recordings, for example, at 3 Hz. So how to deal with all this data. So I'm going to show you first kind of straightforward exploratory analyses, and then I'll show you our tool Rastermap for sorting neurons. So I'll show this straightforward analysis in the context of predictive coding. So this is a type of unsupervised learning. And so in the lab, we've been studying a lot of unsupervised learning recently. And so you can imagine if you're a mouse walking through a forest, you see the same trees day after day, and you might make an internal model of your world of the trees that you see. And then maybe one day you're walking in the forest and you see a Christmas tree. And so you might be surprised. But the question is, what type of surprise is it? Are you surprised because you were predicting a specific tree in the future and you were doing temporal predictive coding? Or are you surprised because this was a new input you hadn't seen before? So I'll show you the results for that briefly, doing simple exploratory analyses. So so Ching set up this task with a virtual reality with 4 stimuli. She trains the mice for a week or so with these stimuli and then tests with other stimuli. So this is an example plot like Kenneth and Celine were also saying, like, you really want to visualize your data as much as possible. So this is an example here of neurons sorted by their preferred response across the corridor. And this is also an important point here is you always want to find your sorting on training trials and plot it on test trials. So we split the recording in half, compute the preferred position on training trials, and this is showing the average response on test trials. And we talk a bit about it in our review here, and Kenneth is also an expert on these sorts of analyses like he discussed too. So basically, we get this sort of picture of what the neural activity looks like to the standard sequence. And then we look at these test images on test days where on a subset of trials, Ching presents these novel stimuli or different stimuli. And so I'll just show you a couple of examples where kind of just looking at this neural activity averaged and neurons by time is useful. So for example, if we put in a novel stimulus here and we look at this plot, we can see this big change in activity for the novel stimulus in this kind of example plot. And actually, even the mouse slows down its running during this time period. So there's also a behavioral readout of surprise in the mouse. When we look at if we replace the C stimulus with a D stimulus on a test day, we actually don't see a big response. And actually, what looks like is happening is the same neurons are responding to this D stimulus that respond to D when it's in the 4th position, which is where it is in the original sequence here. And so it's easy to see this kind of in an example, kind of raster plot of neurons by positions. And in this case as well, the mouse doesn't exhibit behavioral surprise. So you should check out the preprint if you're interested in more details. But kind of a lot of the plots we make in the preprint are of this sort, and we think it's really nice to illustrate the data across many neurons. And the summary is kind of we think that neural and behavioral surprise is related to novel images, not novel sequences or temporal predictive coding. Um, so that was a case where we could actually average over trials. The mouse was seeing the same thing every trial. Um, In this case, Lynn is trained mice to discriminate circle and leaf, and in this case, he actually had the reward timing was random along the corridor. So there was a sound cue at a random position in the circle corridor. It's not followed by reward. In the leaf corridor, it's followed by reward. And this randomness means that we can dissociate reward signals from visual signals. But it also means that we can't just average over trials when we're trying to visualize the neural activity. So I'll just show you what this was the task that I was showing before, the mice running through these corridors, and you'll see them. This is the rewarding corridor, so the mouse will stop to lick here. The mice learn to discriminate these visual corridors very well. So they're licking much more in the leaf corridor than the circle corridor after learning. This is before learning. He also has a cohort of unsupervised mice. I won't show that, but basically just looking at, we have this big plot of neurons by time. We can't average over trials. So instead what we do is we average across neurons. So we put neurons that are correlated to each other next to each other using Rastermap, and then we get a plot that looks like this, and we get sequences during the corridors, and you can see these breaks, and the sequences are actually when the mouse is stopping running. And so we can see those things more easily when we're making a plot of neurons by time here and not averaging over trials. And averaging over neurons here because single neurons are noisy, so we can see things better. So how does this algorithm work for sorting neurons? The first thing we do is we cluster neurons using k-means clustering. So this is another step that kind of helps to reduce the noise. You can also sort single neurons. I'll show examples of that too. But if you have a lot of neurons, you can cluster them first, and the algorithm has the option for clustering first, and that can kind of reduce the noise in the data and makes the algorithm more efficient. After you after the clustering happens, we compute a cross correlation of between clusters. So this is the correlation, for example, for two clusters that are in a sequence, this cross correlation will be high at some time lag that's non-zero, like this here. So we actually take the max over these time lags, and that's what we use for and this is a parameter in the algorithm time lag window. And that's what we use to create this, what we call asymmetric similarity matrix. And so because we're taking this max over time lag windows, it allows us to kind of get the max correlation even across time bins and capture these sequential events like I was showing those sequences in the data. So we sort this matrix so that if this was like a perfect sorting, for example, it maybe would look something like this if it was uh, if basically it had a specific global structure with a power law decay. So we try to get the matrix to look to match this matrix. We also add on this local penalty or matching matrix, which is basically just the first off diagonal is all ones, and you can add that with the various weighting terms. And this is called the locality parameter in the algorithm. And that allows us to kind of overweight for certain data sets that have sequences, we can upweight that parameter to try to bias the algorithm to find sequences. And so the algorithm is using basically traveling salesman-based algorithms to do the sorting, and here's what it looks like when it's running. So you can see the clusters moving around. Basically, it's iterating through many of the possible moves it can make, and choosing the best move that will maximize the similarity between this matrix and this matrix. And then something like this comes out. And after this, so this was just on the clusters. If you do it on the clusters, you might also want to upsample. So we have this upsampling parameter. The default is 10, and basically neurons are assigned to, if we did it with 100 clusters, then we have times 10 possible positions if we have grid upsample of 10. And so there's 1,000 possible upsample clusters that neurons can be assigned to. And then the sorting, I can go back and forth. This is the sorting with the clusters and then the sorting with the neurons themselves. Okay, so that's how we were able to make this plot that I showed you before. Now it's colored by all of the possible kind of behavioral parameters that we have. So the running speed of the mouse, the reward time is this green line here, the tone time. The tone is in both corridors, but it's only followed by reward in the leaf corridor and the lick times, for example. And so actually, and you can see the sequences within each of the corridors that we think are visually evoked because you can see they stop when the mouse stops running and the visual flow stops. Actually, when we were looking at these raster maps, we found this interesting population of neurons that seemed to ramp up to the time of reward and then shut off after the time of the reward. And interestingly, we found these neurons were mainly in anterior regions of visual cortex, more like PPC-like regions. And I can summarize them, basically average those neurons across trials, sorted by the time of the sound cue. So in the circle corridors, without rewards, these neurons are not very active, but they're very active in the leaf corridor, and they seem to ramp up to the time of the reward and then turn off after the reward. Because we think if it's like the reward expectation signal, then after the mouse gets the reward, there's only one reward per corridor, these neurons would shut off. So this is an example of something that we didn't, I mean, we didn't think we would see a signal like this necessarily. And so looking at Rastermap helped us find the signal and quantify it. And we were able to quantify it across different test stimuli as well that Lynn showed. You can also use Rastermap on ePhys data. So this is example data with around 100 neurons from the Buzaki lab. And so this was a rat running in a linear track back and forth, and you can see two sequences for the one direction and the back direction. And one thing Rastermap did here, so this was sorting with single neurons, like I was saying. So the number of the clusters is set to none in the algorithm. What you can see is that the inhibitory neurons, the fast spiking putative inhibitory neurons and the regular spiking neurons are actually separated by Rastermap here, and you can see their different profiles. So this can be a helpful way to visualize your data across trials in ePhys data. Kenneth touched on this as well, that basically neural activity we find is related to the behaviors of the mouse. So this is example of spontaneous neural activity in visual cortex when the mouse is in total darkness. We're not showing any visual stimuli. We've sorted the neural activity by Rastermap. We can see lots of different patterns, even though we're not showing visual stimuli. We can look at where these neurons are with respect to the raster map plotting. That's this color bar here, and each of these dots is the neuron in the sensory motor example window. We quantify facial behaviors, so you can do the motion SVDs like Kenneth described. Recently, Arika Saida developed a method for tracking key points on the mouse's face, and so we can also use these key points to describe different behaviors of the mouse and use those to predict neural activity. Now, more recently, we've been using nonlinear models to go from behaviors to neural activity, and this is what the prediction looks like. So we can capture a lot of the faster timescales of neural activity with the behavior variables that we have. And just to note, in terms of the sorting, so you can look at these behavioral variables and sort your neural activity. So for example, these are the neurons sorted according to their correlation with running speed, correlation to whisking speed, nose speed, and eye area. But a lot of these variables are correlated to each other, and it's kind of hard to see all the diverse patterns if you're sorting in this way rather than using something like Rastermap or another embedding algorithm, where you can actually see populations of neurons that are differentiated. And we see, for example, specific neuron populations like this one here that is quite active just on the onset of running, for example, that's very clear in the Rastermap sorting. You can also use this on trial average data. So this is an example of a task from primates from the Jaizeri lab where the monkeys were trained to reproduce a time interval that they were given. And these are the neurons. This is neurons by time. And this is the trial average response to these different time intervals. And they had these different different blocks with different priors on the average time window. And so you can look, for example, after sorting at what the difference is in the responses depending on the prior in the recording block that they were looking at, the behavioral block. So that could be another helpful way to use Rastermap. And one last example is just showing sorting trials instead of neurons. So this is using Nick Steinmetz's really nice study And I think I might have the same neuron that Kenneth showed from PO that's quite locked to licking. And basically what I sorted here was instead of sorting um, like the trials from a single neuron, I'm actually sorting the trials by features, which is the PCs of the whole recording by the time points. Um, and this is what the raster map looks like after sorting. So this is the example response of this neuron now sorted by raster map, and then for this PO neuron, and you can see the licking that's very periodic and fairly locked to the time of the reward, like Kenneth was describing. And yeah, basically you can look in the Rastermap paper for more details on this. Just like basically one thing we saw was that there are different periods of disengagement, which many other people have quantified in the IBL data, but this was kind of a nice way to just differentiate them. And it seemed to be related to the time in the beginning, whether the mouse was in the beginning or the end of the corridor. And you can check out the GitHub for more details on how to install Rastermap. And thanks everyone for your time.